Hey, this is Dr. Mirup, and Happy New Year. And we are getting a lot of questions about the coronavirus vaccine, and we've been extremely fortunate that a lot of us doctors and our staff have received it. And I don't know if you saw our other videos, we actually have photos and video of us receiving the vaccine. So what I want to talk about now is the actual vaccine. What is it? What's an mRNA vaccine? So I'm going to start off with a diagram and sort of just talk about the coronavirus, and then I'll talk about sort of how the vaccine's made. So we've all seen the spike for the coronavirus. So here's my rendition of the coronavirus. So on the outside, you've probably heard now of people talking about the spike protein. So the spike protein is how the virus binds to your cells. So when people get COVID, what happens is this little spike proteins, they find a little receptor on your cell that's also called the ACE2 receptor. And so then the virus goes into your cell like that. Here's the nucleus of your cell here. So the virus goes into your cell, and there's little spikes here. They trick your cell's machinery into making millions and millions and millions of these guys, of the virus. So what the virus has, if you look at the whole virus, you have this super long stretch of mRNA, and M stands for messenger, and then RNA, ribonucleic acid. So the key is messenger means it's carrying a code, it's carrying a message. Basically, this whole piece of mRNA is code to how to make the whole virus, okay? These nucleocapsids here are just sort of keeping the mRNA in place, and those are also important for other things. So we'll cover that a little bit later for testing and stuff, okay? So, so if we sort of took apart the whole virus, so we have the spike protein, and then we have a real long stretch of the mRNA. So if we kind of stretched it all out, it'd be this big long strand of mRNA. So this has a code. This has a code on how to make the virus. And just for demonstration purposes, let's just say this part of this big long mRNA, that makes the spike protein, okay? This part might make this part of the virus, and this part might do more like how to replicate the RNA. So all these different parts of this mRNA is coding for something different on this virus. And there's a lot more parts to this virus that I'm not drawing here, but that's all part of that code of the RNA, okay? So, so when you get an infection, of COVID, you're getting all of this RNA injected into your cells. So your cells then make more and more of this coronavirus, and that's why some people get super sick and have really bad outcomes with this. The vaccine, so what they've done with the vaccine is they have figured out that this part of the RNA, so I'm just going to draw it a little bit right here. So that is that. All that does is code for this spike protein. That's all it does, okay? So, so the vaccine is really made up of that little piece of RNA. So if we just injected this little piece of RNA into your body, it would disintegrate. It would not do anything. So what has taken scientists a lot of time to learn how to do is to stabilize it. So what they have done is they have found that's actually like it's a lipid nanoparticle. Basically, I like to just think of it as like a fat droplet. And so this fat droplet protects this mRNA from your body from just destroying the mRNA before it has a chance to do its thing. So the vaccine then gets injected, and your immune cells come and they ingest this whole little piece of RNA. So here, let me just draw a different color. So here's that's one of your immune cells. Okay, so so it comes and it gets that little piece of RNA. So your immune cells have a nucleus. and they have ribosomes and all sorts of the machinery here to make proteins, okay? So this immune cell then takes that little piece of RNA, comes in here, and so rather than destroying it, then the mRNA, that little bit of mRNA, starts cranking out a bunch of spike proteins. And that's all it's doing. So it doesn't kill the cell, it doesn't do anything bad to the cell. It just starts making a bunch of spike protein. So then your spike protein goes out into your body, and your body's immune system goes and destroys the spike protein, and then you have immunity. You have antibodies against the spike protein. So that's kind of how it works. Now, usually what you have to do, the vaccines developed now, you have to get 2 doses. Because when you do it the first time, you get a pretty good response, but not a great response. So you get the second dose, then you get awesome production of a bunch of spike protein, and therefore a bunch of not only short-term but long-term immunity. So that's sort of how it works in a nutshell. Okay. So you guys have questions?
So Dr. Meerup, I know we're getting questions on our end, and I know it's just so new.
Yes.
As to, is it reliable? How did they come up with this so quickly? And I know that has been a conversation going on.
Yes, yeah, good question. So this technology of just generally using mRNA into a vaccine kind of started when SARS was coming out. And that was probably about 15 years ago. I don't know exactly, but there was SARS, there was MERS, other coronaviruses. Even the Ebola virus, they were developing a vaccine using this technology. So the technology of using mRNA to do this is not new at all. What took so long to develop, really, to make it work, was the development of this fat droplet that holds the mRNA. Because that is what's unique, and it's a little different between the two that are out now. The Pfizer and the Moderna have a little bit different technology. One, you have to keep super cold, the Pfizer one. So that's really the main difference between those two. But the general framework of using mRNA for vaccines has been around for many years. So what was so awesome is all you need is the code. So back in January, when the virus was first coming out in China, they published a paper with this whole code, and they made that known all around the world. So scientists were able to get on it right from the beginning and figure out which part of that big code made the spike protein, and then they were able to develop this really quickly. So they didn't cut corners. They did all the studies. Another big factor of how it got to be done so fast was funding. A vaccine company making a vaccine without extra funding from the government, they're not going to want to do it so fast. So the fact that they had sort of guaranteed funding for everything really helped matters quite a bit. And finally, to the volunteers to test the vaccine is what it's like, who wants to test it? Gosh, everyone raised their hand. They got 30,000 volunteers so quickly for each vaccine, actually more than 30,000 for one of them. And so they were able to, get the studies done so quickly. The other viruses, and it's good this didn't happen, but you didn't have a big pandemic of the other ones, so you didn't have any volunteers who could potentially be exposed to the virus. So this one, it's unfortunate we had so many people, but the fortunate part of that is that we're able to study this thing so much faster.
And does it alter your DNA?
Does it alter? So yeah, that's a common conspiracy theory kind of thing. So again, this is an RNA virus. It's not a DNA virus, okay? And so there are other RNA viruses that can get into your DNA, but they need, so the HIV virus, for example, I can't really draw it, so it's so much more complex, but two key components in a HIV virus is something called reverse transcriptase. What that does is that converts RNA to DNA, okay? So that exists, but not in this virus, okay? This virus doesn't have any of that. there's another enzyme called integrase, okay? And that's what, if you have some DNA, you have that enzyme, and then you have your DNA, that's what can make it kind of mix in together. So it is impossible for this RNA virus without those two enzymes to incorporate into your own DNA. Sorry, long answer for hopefully then a quick, anyway.
And then I know there's talk about a new strain that was in the UK and,
Yes.
Kind of coming potentially here.
Right, yeah. So basically, the first thing we need to kind of keep in mind is the nomenclature. So a lot of times there'll be a new variant, okay? A variant is different than strain. A variant of the virus means that maybe this little section, and this, and this is different, the RNA is different than the original RNA. But once it's all sort of translated to a new virus, it doesn't necessarily mean the virus is that different. And also, if the variant has nothing to do with the spike protein part of the RNA, well then it absolutely won't have anything to do with affecting the vaccine and how well the vaccine works. It really has other things. And some variants are not good for the virus. So sometimes you have such a bad variant, then the virus really can't replicate or can't bind anymore, and the virus just goes away. So that's an important distinction that you're hearing in the news now, is all the variants is different than a strain. A strain would mean that this virus is shaped or something so different that it's really a lot different.
And I know antibody testing, because I know some people are asking, if I do get the vaccine, can I get the antibody test to make sure that I have enough in my body for protection?
Right. So the current antibody tests that are out there do not check for these antibodies against the spike protein. They only check for antibodies against the nucleocapsid. So the antibodies that we get in these commercial tests are to see if you have antibodies against this part of the virus, not the spike protein. So if you get the vaccine, again, you're not getting all this other stuff. You're just getting the RNA and there won't be little pieces of nucleocapsid either. It's just plain RNA. So that codes for the spike protein. So your antibodies are just against the spike protein, not this. So those tests won't tell you.
And then one last question, because I know, our breastfeeding moms or even moms that are currently pregnant trying to figure out or make a decision on should they get this vaccine.
Yeah, good question. So as far as breastfeeding goes, the consensus amongst all the doctors all across that we've talked to and all of our blogs, our doctor groups and that sort of thing is completely safe. Because it actually will give your child an advantage because, again, you get the vaccine, you make your antibodies against the spike protein. Those antibodies can then cross over to the breast milk, and your baby gets the benefit of getting some anti-COVID antibodies. And so it's actually an advantage to get it if you're breastfeeding. Pregnancy, we don't know yet, really, because on those 30,000 people they got, they're not going to be recruiting pregnant women for obvious reasons for something new. So first trimester, we really don't know, okay? And so I do know a lot of doctor colleagues who are pregnant in their late second, early, and third trimester getting the vaccine. The only thing you have to kind of caution against on timing on that is you do get a Tdap vaccine in your third trimester, and that Tdap vaccine, you have to wait at least 2 weeks, some say 2 to 3 weeks, after you get the COVID shot to get that one, or the other way around. If you got the Tdap, you have to wait 3 weeks. So that's the only thing I would say.
Awesome. Thank you so much for answering all of our questions.
Yes, I'm sure there'll be more, but thanks for listening.